So this quote is pretty appropriate for what we're going to talk about next. So hang on to your hats. Okay, this is I took a whole semester of this in graduate school, and you could easily do like years of this because microbes are so incredibly diverse in how they do their physiology, much more so than we are. I mean, we are amazing organisms, no doubt, but microbes have us beat hands down on how they do their physiology and how they're able to be very flexible and just go on the fly and adapt so quickly to their changing environment. So we need to think a little bit about basically what we're zooming in on today is how do microbes acquire the resources they need to get their ATP synthesized. We all run on ATP. That's what we're doing right now, mainly due to our mitochondria. That's where most of our ATP gets made. There's some other sources too in our cells, but basically the ATP is really ramped up inside our mitochondria. Well, of course, microbes don't have a mitochondria. Where do they put all their stuff to do ATP synthesis, do you think? If they don't have an inside mitochondria, where are they putting it? Some of it's free floating. Where else would they put it? In the membrane, cell membrane. Some of it's in the cytoplasm. We'll talk about that. And then when they really ramp up their ATP production, they actually stick a lot of the necessary enzymes for that in their membrane. So what we're thinking about is how do they take in their resources in order to synthesize ATP? That's called catabolism or catabolic reactions. It's taking in substances, breaking them down, and as you're breaking them down through physiological processes, you create a situation where ATP can get made. When you look at the big picture, like, okay, how is this happening? There's basically three big ideas on how to synthesize ATP. There's this concept called oxidative phosphorylation, substrate level phosphorylation, and then photophosphorylation. We can do some of this, but not all of it. You might have learned about things like glycolysis in other classes. I hope by now, at least, you've heard of that and thought about it a little bit. You might have heard about citric acid cycle, also called the Krebs cycle or the TCA cycle. This is another process where ATP can get made. And then there's the so-called electron transport chain, which is what's really cooking along in your mitochondria. Okay, so when we talk about what's happening in your mitochondria, we use the phrase oxidative phosphorylation. And so if you've taken animal phys, human phys, maybe some biochem classes, we tend to focus on what's happening in humans and other animals. And so you'll hear that phrase, oxidative phosphorylation is going on inside your mitochondria, and that's true. So all of this that I have listed out, microbes can do way more than these examples I've just provided you. Yes, they can do glycolysis and the citric acid and electron transport, but they can do other stuff too. That's pretty fantastic. So what I would like to do is first dispel a misconception that I'm hoping will clarify now that we're in the microbial world. So when you think about this phrase, oxidative phosphorylation, and you hear the word oxidative, I mean, what does that word remind you of? Oxygen, right? Totally get it. That's what I think of too. And so, and it's fine when you're talking about humans, it's fine that you keep that thought. That's okay. But we're not in a human class. We're learning about microbes. When you think about what's going on with microbes, they do their own version of an electron transport chain process. It's not in a mitochondria, but they have a similar idea going on. And they use that to make ATP. And we call it oxidative phosphorylation. But I need you to be aware that when microbes do it, oxygen sometimes is not involved in this process. We do it. We have oxygen sitting at the end of our electron transport chain. But some microbes do that and some don't. And so it's a little bit of a misconception. I don't want you to equate oxidative phosphorylation in this class with oxygen. You have to separate those two. Okay, so in the microbial realm, oxidative phosphorylation should really be thought of as just its electron transport chain linked phosphorylation. There's oxidative phosphorylation, just immediately think about, okay, there's some kind of electron transport chain. Maybe oxygen's involved, maybe not. We clear on that? Okay, this is a big deal because when I ask you questions in previous examples and previous semesters, and I ask about what's going on in the electron transport chain, it's so easy for students to just knee jerk back to it's oxygen. And it's like, well, yes and no. In this class with microbes, yes, sometimes, but some microbes, they don't want anything to do with oxygen. They will die in the presence of oxygen, but yet they can still run an electron transport chain and get ATP. So what are we talking about here? This is a mock-up of how a bacteria might do electron transport chain processes. And you can see that it's in the cell membrane that they're pointing out. And you can see that there's all these proteins and enzymes embedded in the cell membrane. And so here, I'm not going for the details. Like, I don't want you to memorize every player in the process here. You do that in biochem classes, and that's fine. But in this class, I want the big concepts. I want you to understand that there are chemicals coming into the electron transport chain process, and they donate off electrons. So that's what they're trying to show here with NADH, FADH2. Those are giving electrons. So the red line, imagine electrons being donated on down the line from each donor and acceptor that's embedded in the membrane here. Okay, so that red line is representing electron flow from donors to acceptors. And then you see oxygen here at the very end of the electron transport chain. And so they finally show, 2 electrons are coming in here, and it will react with oxygen. And eventually, it's actually 1 electron at a time. It's not really 2 at a time. What happens is 1 electron at a time will get to oxygen, and eventually you'll get enough that you'll create water. Now, the goal of the microbe is not to create water. That's not their goal. That's the final product being shown, but they don't really care about that. What they're doing is setting up a situation through this electron transport chain process that allows for ATP synthesis. So anytime you start learning about electron transport chain, you should start immediately thinking about, oh, I wonder if there's a proton gradient attached to this idea. And yes, there is. All right, so here you see as electrons are flowing from left to right here, all the way to oxygen, you see something else going on. You see these protons getting pushed across the membrane at key spots. So what they're trying to show here is that you start to get a buildup of protons outside the cell, less protons inside the cell. So now you have a proton gradient. Anytime you hear about a proton gradient, that's potential energy. That's something the microbe can work with in terms of, I can use this potential energy to do something I need to do. And it can be a variety of things, but one of the big things they have to do with that proton gradient is make ATP. So that's what's being shown here with this ATP synthase. It's this amazing little molecular motor. They actually refer to it as a molecular motor, that these protons that have built up will want to diffuse across the membrane with the laws of diffusion. As they start to do that, they'll have to go through this ATP synthase because they're charged molecules. They can't just fit through this very hydrophobic section. So they're going to flow through the ATP synthase. And as they do that, creates a situation, a torque force, where it allows these ADP and the inorganic phosphate to react. And then you get ATP. So this is showing oxidative phosphorylation with oxygen involved, but it's not always oxygen involved. So oxidative is referring to the idea that you're stripping electrons. I like to use that phrase, stripping electrons off of molecules and sending them into a transport chain process. If you oxidize something, you're getting electrons from that substance. That's really what it means. Doesn't necessarily mean oxygen's involved. You're just stripping electrons. So you're stripping electrons off, sending it through this chain, creating a situation that allows for proton pumping across the membrane, getting your proton gradient, which then gets you your beautiful ATP. That's what the microbe is going for. We call it oxidative phosphorylation because right here, you are phosphorylating ADP. You're adding a phosphate, so you're phosphorylating the ADP. You with me? Okay, yes. You don't want us to. Oh, no, no, not in this class. I asked for a little bit more in Gen Micro, but not for this class because you need the basics of phys for this class. You don't need the details because I need you to apply what you're learning in physiology, this section, to better understanding how microbes live where they live, how are they adapting, why don't they live here, how come they're only there? And so it really does help explain the world of the microbes and how they live their life in these different habitats. Okay, so yes, many microbes can do this. You'll often hear about aerobic respiration, like we're doing that right now. That's what's happening in our mitochondria. Aerobic means oxygen. We're using oxygen in our mitochondria as the terminal electron acceptor, similar to what you just saw. And that's great. Oxygen gives you, if oxygen is the last player in that transport chain process, it gives you a higher ATP yield than any other process, like a fermentation process or something like that. Really good ATP yield. And that's because, without going into huge detail, oxygen is the best electron acceptor on the face of the earth. We don't know of a better one. It's so good at accepting electrons, and you get so much free energy from that process to make ATP. It's the best one we know of. So if you're a microbe and if you're not poisoned by oxygen and oxygen's around you, you're going to want that oxygen. You're going to want to do what we just went over on that previous slide. If you're smart and savvy, you would do this because it's going to get you the most ATP. But not all microbes live in environments where you have oxygen, and yet they live their lives just fine. Okay, so here's another portrayal of an electron transport chain in a microbe. You can see there's quite a bit going on here. It's complicated. It's actually pretty amazing, but this is a little bit better showing the electron flow idea. You can see the black lines with the E on there. And so you can see the NADH coming in here, donating off electrons at the EH2, right? And then it flows through all these different donors and acceptors, passing the electron on down the line. And then you can see oxygen sitting at the very, very end. And then eventually it will get reduced to water, but that's not the goal. The goal is the microbe is setting up this very neat situation where all of this flow is creating some potential energy, this electron flow. It does create some potential energy to allow for proton pumping. And then you get your nice proton gradient from that process. So what we are doing is something similar to this in our mitochondria. Microbes are doing it on their membrane. This is a very big picture idea of how some microbes live their life in terms of how they're getting their ATP. And we do something similar to this. Right now, if you had a nice breakfast, hopefully you are, your body, your cells are breaking down the carbohydrates into eventually glucose, and your body is running glycolysis. And microbes do that. They do this primarily in their cytoplasm. And you see, you do get a little bit ATP. Not much. You only get 2 with per glucose coming in, but it's better than nothing. And then what I like about this image is it's showing how these physiological processes are all linked together. So here in the middle, you're seeing this after glycolysis is running, you get a shift over here where you get pyruvate coming into the TCA cycle or the Krebs cycle. And your body's doing that too. And microbes can do that as well. And you can see as they're running that cycle, they do get some ATP produced. Not much, better than nothing. And then you can see some of the products from the TCA cycle. We've already been talking about them, NADH and FADH2. Those get fed into the electron transport chain or electron transport system, into the cell membrane. That's where all the proteins and enzymes are embedded. And so that's where they're saying, yep, oxidative phosphorylation is going on. And they're calling it oxidative because they're using the example of oxygen as the terminal electron acceptor. So this image is nice because it's showing you the flow of, you do something similar to this, you're doing this right now, microbes can do this, right? And it's all linked together, and it's a very beautiful physiological tapestry of glycolysis, working on glucose, getting it to pyruvate, that gets sent over to the TCA cycle, that creates some NADH and FADH2, which gets fed in to the electron transport chain. And again, the goal here is to create situations where you can get ATP synthesized throughout these processes. And you can see the difference here. Oxidative phosphorylation, 34 ATP instead of just 2. A big difference. So if you have oxygen and you're not poisoned by it, you should use it. ATP is amazing. It's a little molecular motor, many subunits that actually rotate and create a torque force that allow for the ADP to get phosphorylated. So oxidative phosphorylation. And when you phosphorylate ADP, of course, that gets you that lovely ATP. Chemical energy that we all thrive on and have to have. Okay, so going back to this. So oxidative phosphorylation, that's the electron transport chain process. Yes, oxygen can be involved, but not always. This next idea of substrate level phosphorylation, microbes can do that. You're doing it right now. And this is the idea that an excellent example of it is actually glycolysis. When you do your glycolysis, you're running a fermentation reaction. So substrate level phosphorylation. This happens in the absence of oxygen. Oxygen's not involved anywhere in this process. Okay, so you can do it in an anoxic environment, no oxygen. AN on the front of a science word means without. So without oxygen, you can say they're anaerobic microbes, and they still can manage to make ATP, but it's at the substrate level. So what they're saying that there is, during the process of glycolysis, when you have all these chemicals, in the physiological pathway, it's at those substrate chemicals that you start to see some kind of phosphorylation event where ADP can get phosphorylated. So basically what we see is that there will be a substrate in the pathway, like in glycolysis, that has a phosphate attached to it, and then it'll donate it off to ADP. And that will get you your ATP. So when that phosphate gets cleaved off, it's a little bit of an energy push, so to speak, in order to get it to attach to ADP to get you your ATP. Okay, so you're still phosphorylating ADP. It's a phosphorylation, but it's at the substrate level. So a simple mock-up of that is here. Here you have substrates in a pathway, physiological pathway, ABCD. And you see a phosphate coming in and phosphorylating substrate B. And then you see that you get this transfer to substrate C. And then here you see that substrate C is going to go ahead and donate phosphate to ADP to get you your ATP. So it's at the level of the substrates where you see this transfer of a phosphate to ADP. So that's another really big idea in the microbial realm. A lot of microbes do this. And it doesn't give them huge amounts of ATP, but it gives them enough to survive and do some modest growth. So again, this is glycolysis. No, I'm not asking you to memorize this. I want you to think about the big idea here, where you can see that there's phosphate. The purple P here are the phosphates attached to these different substrates. And you can see that there's key points in the pathway where you get the transfer of the phosphate off of the substrate on to ADP. And so that gets you your ATP. So you're doing this right now. Lots of microbes do this. And if, the whole beer and winemaking industry really rely on microbes doing this because it gets them the ethanol that they're going for. Right, so lots and lots of fermentation processes are, microbes, that's probably the most famous one that humans know about, right, is ethanol production. Probably I'm sure a multi-billion-dollar industry worldwide. So we've certainly capitalized on yeast's ability to ferment and end up with ethanol. But you can see here that there's other stuff, other pathways, other options. And it really does depend on the microbe, what you feed it, what's it adapted to, what kind of habitat is it from. And so here's another image just trying to impress on you that there's lots of options available to microbes. Microbes don't just make ethanol when they ferment. They do all kinds of things. And so here you're seeing all kinds of different products being made, all kinds of different pathways, all kinds of different starting substances. Doesn't have to be glucose. It can be other molecules, even amino acids. Okay, so it doesn't even have to be a sugar in order to run fermentation. Here's another image just trying to impress on you how diverse fermentation can be. And it just depends where the microbe is living and what it's adapted to, what kind of substrates it wants to bring in and run. Here, this is showing some of the uses we make out of it, in the human world, where you can, of course, see the alcoholic beverages, but there's lots of other options here that in industrial microbiology that we still make the microbes do this. It's not like something that we synthetically make. It's still grow up the microbes in huge vats and feed them the right substances, give them the right conditions, and you can get them to ferment these different products that you're going for. Lots of options. Okay, so very, very diverse. Yes, microbes do glycolysis like we do, but they do so many other things that it's kind of mind-boggling. Here's just some other fermentations. Again, you can see a range of different bacteria. There's all kinds of, just limitless, it seems, amount of bacteria that can do some kind of fermentation. And I'm not going to ask you to memorize this table or, spit back this kind of information. I'm just trying to get you to think about, it really does depend on where they live, how they've adapted to their environment, what kind of substrates are they faced with as a source of nutrition. And then that will help you start thinking about, well, maybe they're doing fermentation, maybe they're using oxygen, maybe they're not using oxygen. And so with fermentation, though, you don't have oxygen involved in here anywhere. So if you look at any of these reactions, nowhere in there is oxygen a player. Okay, so absence of oxygen. There's another way that you can see microbes working their physiology without oxygen besides fermentation. And this is called anaerobic metabolism. So this is another strategy. And some people call it anaerobic respiration, which sometimes is confusing to students because it's like respiration, but that's what we're doing. We're sucking in oxygen. What do you mean? And so I tend to use the word metabolism instead because it's without oxygen, anaerobic, without oxygen. So what we're saying here is that there is an electron transport chain process going along in the cell membrane of the microbe, but no oxygen is at the end of that electron transport chain. It's something else. It can be an inorganic molecule sitting there. It can be organic molecule. They have lots of options. But it's still this typical idea of an electron transport chain process where you have several proteins and enzymes embedded in the cell membrane, and you have that electron flow from donor to acceptor on down the chain until it reaches a final electron acceptor. It's important that you realize that if they do anaerobic metabolism, not as much energy is released in that process compared to if they had oxygen. And so the consequences of that is that they can't make as much ATP when they run their electron transport chain. Because remember, oxygen is the best electron acceptor. You get the best ATP. Now, this will get you some good ATP, just not as good as if oxygen was there. And again, the microbes don't care about the water, or, well, they don't really make water with anaerobic, but some of the products that get made with anaerobic, that's not what they're going for. You know, the very end product of the chain process, they don't care. They just excrete that. It's a waste product to them. And that's important to think about. Same with fermentation. When yeast ferment and we have them make ethanol, they don't hold on to that, right? They excrete it out and then they purify it and bottle it and sell it. In fact, it's a real challenge for the alcohol industry to find yeast that can handle their waste alcohol, the ethanol. It poisons them. It's waste. You know, it'd be like us swimming around in our urine. Eventually it's going to kill you. And so trying to find yeast strains that can handle high percentage of alcohol in the solution they're swimming around in has been like, you know, the holy grail of the beer and winemaking industry. And they made strides, but so far there's like an upper limit. You can't push it above that. So, but that's an important concept, because when we start talking about microbial habitats, and I've already said this more than once in this class, you know, one person's trash is another person's treasure. Same going on here. They're excreting these products because they don't care about those products. They just care about, I created a situation to make ATP. That's what I wanted. Okay. So if you look at anaerobic metabolism side by side, on the left, you see aerobic with oxygen. On the right, you see anaerobic, nitrate. A nitrate. This is a favorite. A lot of microbes love to use nitrate. That's usually fairly abundant, especially in soil systems. That can be pretty abundant. But if you compare the two, I think you can see that it's still, the one on the right is still an electron transport chain process. You still have electron flow from donors to acceptors. You still have protons getting pumped across the membrane to establish a proton gradient. But again, whether it's water being produced, like you see on the left, or in this case, it's nitrite being produced on the right, they don't care about that. They'll just excrete that and call it, you know, that's fine. I don't care about that. What I care about is that I made a proton gradient. And the artist drew this. If you count up the protons, it looks like there's 5 on the left and only 4 on the right. So the artist is trying to, you know, show that you don't get as great of a proton gradient. And that means you don't get as much ATP. Okay, so that's what they're trying to imply. The one thing the artist failed to do, which kind of drives me nuts, but they have this lovely depiction of an electron transport chain, but they forgot to link it to ATP production. All right. So I put this in here to kind of emphasize the point. You can't divorce the two. All right. If you see an electron transport chain process, look for a proton gradient, and then immediately look for ATP synthase sitting there, ready to have those protons flow through it. And then you end up with ATP. Okay. So proton gradient. How do we make that? Well, it's through this electron transport chain process. And then ATP synthase, well, that's how the protons diffuse back. And in that process, they're phosphorylating ADP. Okay, so here is an example of a more complex electron transport chain process. So what we just looked at in the previous one stopped right here, where you had nitrate being made. And then that got excreted by that microbe in that previous example. Here you're seeing this particular one said, well, I'm not going to excrete that. I'm going to keep pushing it further into more of an electron transport chain. So what you see here is it just keeps going and going and going and going and going and going until you end up with N2O. Is that nitrous oxide or nitric? I can always forget those two. As the terminal electron acceptor. And then the end product is N2, dinitrogen gas. So as they're doing this, you can see there's proton pumping going on at various stages, which is great because, and again, the artist left off the ATP synthase. So imagine that proton gradient developing and then flowing through the ATP synthase, those protons, to get you your ATP. So Pseudomonas is famous. Many species of Pseudomonas can do this. This is Pseudomonas stutzeri, which has been pretty well studied. And so what it's doing is it's taking nitrate here in the pink, NO3 minus, and that accepts an electron, which gets reduced to nitrite. And then you get nitric oxide, NO, and then nitrous oxide, N2O. So the nitric oxide, nitrous oxide, and then dinitrogen, N2, those are all gases. So now you're starting to get a feel for, oh, N2, we got 80% of that in our atmosphere. Well, it's because of microbes like this, over billions of years doing something like this, that has allowed for dinitrogen gas to build up in our atmosphere. These other gases are also coming out into the atmosphere. They just excrete them. So this is a way for nitrogen, microbes are, recycling nitrogen. And so some of this could be happening in your Winogradsky columns right now. This might be happening. Some of the microbes might be doing this. And so tomorrow, I don't know if you'll see it tomorrow, but what you should be looking for every week when you go into lab, look at your Winogradsky, look for bubbles. You might see some bubbling going on, released from the soil, getting up into the water column. And some of that could be some of these gases. So this is a way that we see microbes recycling nitrogen. They take some microbes have the ability to grab dinitrogen from the atmosphere and turn it to ammonia. And then there's other microbes who kind of do the, well, I'll add dinitrogen to the atmosphere. And so you get this cycling going on of the nitrogen compounds. So people who work with soil and growing plants, this is important. You want to know any microbes in your soil, what are they doing in terms of nitrogen? Because if I am depending on growing microbes for a livelihood, I don't want denitrification going on. That's not what I want. So I would take soil samples and look for denitrifiers. And if I have an overabundance of denitrifiers, I try to do something about that. Because plants really love nitrate. And so if you have competitor microbes in the soil near your plants and they're like, well, I like nitrate too, I'm going to take it, then my plants don't have enough. So you don't want this going on in your soil if you're trying to grow plants. And so knowing a little bit about the microbiology and the physiology of what they're doing in the soil system, you can try to tweak that to your advantage so that your plants will have an easier time of thriving and producing a better yield. Questions about that process at all? Or anything else we've been zooming through?
Why wouldn't you see more ethanol production from anaerobic microbes if like aerobic microbes use glycolysis too?
So So when they use glycolysis, you're asking why you don't see more ethanol production?
Yeah, I guess like if you think about ethanol production, it could be like anaerobic microbes, like the,
Mhm.
like nitrogen is their like terminal receptor.
No, with ethanol production, when you see like in the brewing industry, when they take yeast and say, you have to make ethanol for us. What they're creating is a situation where they make the microbe do glycolysis essentially over and over and over and over and over and over and over. And so they, if you've ever done beer tours in the backs, they have those huge copper vats. I don't know what the official word is, but they will create an anoxic environment. They'll feed in a lot of sugary substances. And they'll actually, well, they do a lot of bit tweaking to really fine tune it so that there's lots of lovely sugar that the yeast can have immediate access to. And then they just keep making sure, I'll keep feeding you glucose, the yeast just keeps doing this over and over and over. And they keep spitting out the, ethanol as waste products. And so they force this situation. And so they basically are turning off the yeast's ability to do any of those other pathways we've been talking about. Yeah, yeah. And so that's how they can get some good yield of ethanol to a point, and then the microbes start swimming around too much in its own waste. It's just too much. And they get poisoned off. So they die if they keep it going. So. Other questions about these processes? Don't panic. I'm not asking you to memorize this. Okay, that's not what I'm asking. So if you're unsure when you're studying how deep to go, just ask me. I'm much more concerned that you get the theory behind it, because that really the light bulb starts to go off when you start then applying this information to microbial habitats, which is what we're going to talk about next, in the next section. Okay, a couple of other things we need to think about. And I took a whole semester of this in grad school, and then I took further classes beyond that. First semester with my major advisor was microbial phys guru. And so, I mean, I'm just touching the surface here on what's capable. So, but I do need to give you a couple of other ideas to think about. This is a big one. This is a pretty amazing chemolithotrophy. What that means, chemo just means chemicals. They're using chemicals. So sometimes we just drop that. We just drop that phrase off and just say lithotrophy. Litho, if you look that up, it means rock. So they're trying to get across the idea of inorganic molecules are being worked with here. And then troph means to use. So inorganic compounds, CPDS is compounds, are being used. We can't do this. There's no way we can do this. All right. And so they're actually using them as electron donors. They're saying, they find an inorganic chemical and they say, ooh, you have some electrons that I can take and use and put that into my electron transport chain process. They also can use inorganic compounds as a source of energy. And I'll show you some examples. Where are they getting all these inorganic compounds? Well, they're all over the place. Believe it or not, if they want to use hydrogen sulfide, one volcanic eruption provides a lot of hydrogen sulfide that will then get dispersed globally. Lots of inorganics found in agricultural fields with the inorganic fertilizers that get put onto the soil. Mining processes, there's all kinds of options there. Just our burning of fossil fuels creates inorganic chemicals that the microbes can use. All kinds of industrial waste products that we've spewed out into our lovely environment. And then just my other microbes doing their thing can produce inorganics that then a nearby neighbor would say, oh, I like that. I'll use that as an electron and energy source. So inorganic means there's no, it's not like glucose where there's lots of, there's no carbon. But all organisms that we know of on the face of the earth are carbon-based. And so they have to get their carbon somehow. They actually can grab it from carbon dioxide. We can't do that. But carbon dioxide is inorganic. That's not an organic molecule. Okay, CO2 is inorganic. Some kind of hedge their bets. They grab electrons from inorganic, but they're not quite good enough to use carbon dioxide as a carbon source. They didn't evolve that ability. So they rely on some carbon-based substance near them, like maybe decaying leaves that are releasing some carbon that they can snag and take. So heterotrophs, we're heterotrophs. We have to eat something that was once living that's organic in order to get our carbon. So this is really bizarre. I mean, this is so different from what we do. We can't do this. By the way, if they are heterotrophs, where they have to get their carbon from some organic substance, but then they grab electrons from inorganic, so they're kind of a foot in each world, we call them mixotrophs. They kind of mix it up. So what are some examples? Here are some examples. And again, I'm not asking you to memorize this, but if you can provide an example. So if I say to you on the upcoming exam in a couple of weeks, hey, you know, what's an idea of lithotrophy? You could say any of this and it would be fine. And so the top line, hydrogenotrophy, I'm saying that word, basically they're using hydrogen gas, H2. And that's inorganic, and they're taking electrons off of it. And you can see here, they like to use oxygen as the terminal electron acceptor. So this is still an electron transport chain process that they're running. And it depends who they are and where they live. They might choose to use oxygen at the end of their electron transport chain, but not always. Sometimes you see some other options. We're going to talk about some of these in more detail, but this is just trying to give you some idea. I would like you to be familiar with hydrogenotrophy as a really good example of inorganic. And the reason I'd like you to know that in a little bit more detail is that hydrogen is just so good at donating electrons. And so it has the fancy way to say it is they have sufficient reducing potential to donate electrons to nearly all biological electron acceptors. So it's just super good at donating electrons. And when they do that, when microbe takes an electron from hydrogen, it creates a situation where you get some really good ATP. And so here you're seeing this idea of they're stripping electrons off of hydrogen in this top reaction. They put it through an electron transport chain process, oxygen sitting there at the end. And you get some good ATP. So here is what it looks like. Just 1 second more, please. Here's hydrogen on the top, getting stripped of electrons, being sent through an electron transport chain process with oxygen at the end. And you get your ATP. So it's still an electron transport chain process. It's just that they're grabbing some inorganic molecule and stripping electrons out of that. We can't do this. Lots and lots of microbes do use hydrogen. If they have hydrogen around them, they'll grab that right away. That's like top pick. But if they don't have hydrogen, they can use some other things. Okay, thank you for your patience. We'll stop there.